All right, thank you so much to the organizers for having me. Thank you all for staying. Hope to make it worth your while. I am Julie Jurgens. I'm a representative of the Knowledge Portal Network at the Broad Institute. So our team works to build open access resources to make research more accessible in multiple different areas. And I'm going to be talking about one of those resources that I'm really excited about today. That is the Common Fund Data Ecosystem, or CFDE. The NIH Common Fund is a funding mechanism to support multi-center, multi-institutional collaborations for research. And in doing this, it catalyzes discovery and aims to address major challenges that face not just one, but multiple branches of research. It programs that accelerate emerging science, remove some of the roadblocks to research, build up the research workforce, and support high-risk, high-reward science in order to change the trajectory of biomedical research. Common Fund programs are extremely variable in terms of what their subject areas are and what they do and the types of data they collect, but these are some common themes among them. They transform science and discovery, build catalytic data resources, and re-engineer the research enterprise. I'm sure you've heard of some Common Fund programs already. Examples are Kids First, which we just heard about from Jared, as well as GTEx. So anybody in the human genetic space has probably heard of this. It's a staple for looking at gene expression. But there are many others that you may or may not have heard of. For instance, HubMap, which is developing single-cell atlases of the human body. Glygen, looking at protein glycosylation. Or Metabolomics Workbench, looking at metabolism and how metabolomics can understand various processes. These resources, as you've just heard, are quite wide-ranging in terms of the types of data that they collect. Some of these are common among various programs, so you can see several CFDE programs, for instance, share transcriptomics or other types of omics data. But in other cases, these data types are very individual to each program. So for instance, you can see here Spark, one of the CFDE programs that focuses on the nervous system, has multiple types of microscopy imaging, phenotyping, electrophysiology, and other data types that are not shared with many of the other programs. So you've heard about the Common Fund, you've heard about the programs within it, and now I'm going to tell you about the Common Fund Data Ecosystem, or CFDE. CFDE aims to take multiple Common Fund programs and integrate their data and resources through a cohesive ecosystem. In doing this, it promotes accessibility, use and reuse, and integration across these entities to form novel biological connections. So from what you've heard, CFDE is extremely powerful and has a lot of potential for discovery in human biomedicine. However, this is hindered by a familiar problem that we've heard about 2 other times at least in today's session, and that's data silos. So these exist between CFDE programs as well as between CFDE and other external initiatives. The obvious solution here is to bridge the silos, right? But that's easier said than done. Each program has its own unique data types made with their own use cases in mind, their own ontologies, and in some cases, bridging these gaps can seem almost impossible. One solution is to promote use of a standard ontology and to harmonize across the programs. But another solution would be to find common types of data that are already inherently interoperable. I'm going to give you an example of one of those today, and that's gene sets and how they connect to human phenotypes. So gene sets, as the name implies, are just lists of genes. These can be generated from a number of different experiments, for instance, differential gene expression. And because of that, they're shared among many different CFDE programs. So they're a means of connecting within our ecosystem. They're also generated by many other external sources, so they're a means to connect to these other ecosystems. And they provide a way of translating beyond just an individual gene to actual pathways and mechanisms that people care about for understanding human disease. Today, I'm going to tell you about a tool that we've developed called the CFDE GeneSet Browser. This enables users to connect cross program analyses of gene sets and how they relate to other genes, human phenotypes, and mechanisms. The GeneSet browser draws currently from 6 different CFDE programs, connections between those programs, as well as individual analyses that we performed on these programs' data, such as differential gene expression. Questions that this browser enables you to ask are which gene sets capture important mechanisms for disease, and which of those mechanisms are actually relevant to human health. The GeneSet browser leverages a method called Pigeon, priors inferred from gene annotations. Pigeon allows users to estimate the probability that a gene is involved in disease using 2 major lines of evidence. The first is GWAS support, and the second is GeneSet support. So I'm going to give you an example of what each of these means. Let's say you're looking at the FTO gene, and you want to know how likely that is to be involved in body mass index. You can start with GWAS support and see that human GWAS in multiple independent studies have implicated the FTO locus in BMI. However, if you look outside of human GWAS, you can also see that multiple gene sets, for instance from mouse models, also implicate increased FTO expression with increased BMI. So the collective result of these 2 lines of evidence shows that there is very strong support for FTO involvement in BMI. Pigeon integrates nearly 7,000 multi-source common and rare disease traits. These are drawn from the Association to Function Knowledge portal and GWAS catalog, which study common disease, as well as from Orphanet, which studies rare disease. I invite you to follow along with me here. This is a revamped version of our CFDE GeneSet browser that is just coming online now, so I encourage you to try it out. You can access it through this QR code here or through the web link that's shared at the bottom of the screen, and I'll have that up on some of the subsequent slides as well. To start, I'll take you through the search interface. You can select what you want to search for, be it an individual gene, a couple of genes, a phenotype, CFDE program, or a gene set ID. And here's an example of the types of results you can get. Let's say you search for an individual gene, FTO. This interface allows you to estimate the probability that that FTO gene is involved in various different human diseases. And that's again using those lines of evidence we talked about before, GWAS evidence, gene set evidence, and the combination of the 2. Here on this plot, you're seeing the combined scores of both GWAS and gene set evidence relative to a whole host of human traits, common disease traits and rare disease traits here. In the table of results below, you can see the top human trait associations, and as expected, body mass index comes up. If you click the show button over here, you can get more information about each association. So in this case, you're seeing, In this case, you're seeing the individual gene sets that actually contribute to that BMI FTO association through Pigeon. This is another search example. Here we're searching by a CFDE program. In this case, MotorPack, a program that studies the molecular effects of exercise. Through this type of a search, the output that you get is an estimate of the effect that each gene within the gene set is involved in disease. So these estimates are available for the gene set in isolation. We call that the marginal effect, or for the gene set considering all the other gene sets that have been tested. We call that the joint effect. Here you can see the result for MotorPack, and many of these top results are for various reticulocyte percentages, counts, and other reticulocyte traits. And that's expected since this is a known association in exercise. Again, if you click on the show button here, you can get more information, in this case about the actual genes within that gene set that are driving the association and their relative contributions to that association. This is a breakdown of what this actually means. So for this particular example, you're seeing that this set of genes, upregulated in blood after 2 weeks of exercise in rats, is biologically interesting, that the genes within that gene set have roles in the trait that you're looking at, reticulocyte count, which specific genes involved in the gene set are involved, and finally, that the genes have a causal role in the nature of the genetic variation. If you want more information, you can click on the hypothesis button here, which will give you an LLM-generated hypothesis about why this connection exists. So in summary, I've shown you the current capabilities of the GeneSet browser so far. It identifies known and novel connections between sets of genes, mechanisms, and human phenotypes. It also connects multi-source data types and phenotypes from both CFDE and other external sources, and leverages a common vocabulary of the gene set and of the human genetics backbone. So it supports interoperability and AI/ML readiness. So that's all great, but we're still left with some problems, right? First, users receive many, many results on our interface currently. So it's hard for a user to know which is most relevant to me and what I actually care about. To address this problem, again, we piloted a brand new feature available through this link here or through the URL shown here. This is called semantic-based searching. So the idea is that instead of searching by just a keyword, you can search based on the meaning of what it is that you're trying to identify. I'll give you an example. Let's say you're interested in apoptosis. You start typing APOP and might come up with an autofill in a standard search, giving you apoptotic signaling pathway. Other program has program cell death as the actual name of that entry. It would not come up in a standard search, even though it essentially means the same thing that you're looking to search. So what if, instead of using that standard sort of keyword-based search, you could actually embed these two terms into a vector and perform a vector-based searching through. This is what we've done, and I'll show you an example of an output. So let's say you want to search for a term like abnormal glucose uptake. You can write that in free text in your own words, and the program will come up with the top CFDE phenotype and gene set associations that are significant as determined by our pigeon algorithm. It'll also come up with the top CFDE phenotypes that are actually related to that based on semantic searching. So you can see in this case, some of the top terms do have the word glucose in the name, but others don't. For instance, there's one about carbohydrate metabolism here. So these are things that are enhancing the capabilities of this interface to address the actual user needs. So what's next? We want to move beyond text embedding and to allow users to connect independent concepts and phenotypes. The way we plan to do that is through agentic AI. Imagine a world where a user could come in, enter by free text what it is they actually are interested in studying, and get an output. So this is an example of a beta version of this that we're developing out and plan to release hopefully in the near future. In this example, the user says, I'm a biologist interested in the interplay between metabolic disorders and neurodegenerative disease. Tell me what gene sets and biological mechanisms are impacted in both these diseases and how I can study them to come up with better therapeutic targets or knowledge. Here's an example of the output. And sorry, it's a little small on the screen, so it might be hard to read, but the summary that is provided actually gives you shared gene sets and biological interpretations between those two diseases. So this is something that the interface on its own currently would not enable, but that this connection provided by agentic AI would. So in summary, we can use newer technologies like text embedding and agentic AI to further evolve our interface and make it more customizable for user experience. In the future, these are some additional plans that we have for our gene set analyses. We want to better curate, standardize, and integrate new and existing CFDE gene sets, streamline the user experience to make things both more used and useful, synthesize these results into interesting scientific findings within CFDE and beyond, validate some of those findings through experimental collaborations, and extend into new target communities. So we'd love to hear from you. If you have a chance to come talk to me, let me know what types of tools and resources are actually needed to accelerate your research, and how can we make CFDE more accessible and useful. Finally, if you want to learn more about CFDE, you can do it right here at ISMB. So these are a couple of other sessions that we have coming up. On July 23rd at the education track, you can learn how to use the CFDE training center's tools to build omics skills. And we have multiple talks at the Elixir NIH ODSS session, also on July 23rd, where you can hear about Metabolomics Workbench, Glygen, HubMap, and Senate, some of our CFDE programs, and the amazing advances that they're making. So huge thank you to our incredible team who worked really hard to bring you the tools you're seeing today. Big thank you to NIH for our funding, and to all of you for staying to listen with me today. Thank you.
Thank you, Julie, and yes, thanks for staying. I think we're at break right now until 4:40. If folks have questions for Julie, or maybe even for Jared, if you're sticking in the room, we can chat, but